Hi, everyone. Thanks for coming. Really excited to have a talented scientist and a good friend here, Dr. Nagissa Mahmoudi. She's fairly recently tenured associate professor at McGill University. So for any students out there during lunch, it might be nice to talk to her because you may be thinking about your next steps. And I can say that she's someone who is very productive, very talented, but also a good person. It will tell you what you need to hear. We had a conversation last night that we're lucky in our field that good people also do good science. That's not always the case, so she would fit in well here. So Nagissa got started studying integrated biology at the University of Toronto. And then she moved south of the border and then went back north of the border. And we met along with Greg Hanks while we were all basically postdocing with Ann Pearson at Harvard University. And I was very lucky to work with her because she taught me so much about microbiology. And also, my job was to build a bioreactor, and I was incapable of doing it because I had surgery on my hands. So she was not only the brains, but she was the machine that made this thing work. So I really don't know what my role was there, other than to be in the presence of wonderful. And she's continued using very interesting and novel techniques, both in the lab and in the field, to make some very nice contributions to our understanding of microbial processes and the reactivity of organic matter and sediments. And so I don't want to take too much of her time, let her speak for herself. And today she's going to talk about picky eaters, combining, I guess, some of her more recent work with some of her past work.
Yeah.
So welcome, Nagissa.
Awesome. Thank you, Steve. So let me flip this on. Okay, can you guys hear me in the back? Yeah. Okay, fantastic. Thank you, Steve, for that introduction. So Steve says I was the machine. I will show you a picture of what we worked on. I would call myself Steve's shoddy apprentice because I was not that good at building stuff. But Steve taught me the difference between an ultra torr and a swage lock fitting and how many times to turn it. And so I will get into some of that today. So I'm a geomicrobiologist, and my group at McGill studies, sorry, I forgot I have to switch two things. So we study what microbes are essentially eating in the ocean. We're interested in how they select and access carbon substrates. And we study this from different perspectives. So today I'm going to talk to you about some of the work we've done to understand this in the context of modern marine environments and more from like a microbial ecology perspective. And then I'm going to finish the seminar with some work we've done to think about this in past environments and thinking about the evolution of the capabilities of microbes to break down organic matter and how that's changed through Earth's history. And then lastly, I just wanted to mention we do have some projects thinking about life on ocean worlds. I'm not going to talk about any of that stuff, but if it does interest you, then you can talk to me after or email me, and hopefully it'll be a starting point for some discussions. Okay. All right, so I wanted to start up with this really simple diagram of the marine carbon cycle. Maybe it's not needed for this audience. This is very oversimplified. So if you study zooplankton, I apologize, or maybe just cover your eyes for this slide. But we broadly know how the marine carbon cycle works, right? We have CO2 that crosses over the air-sea interface. It gets consumed by phytoplankton, and they turn that CO2 into biomass or particulate organic matter, which is really just sort of decaying biomass. And then most of that particulate organic matter, it gets broken down in the water column by, usually bacteria and archaea. They respire it back out as CO2. And then a fraction of it continues to sink down into sediments, where microbes and sediments continue to process it and release CO2 and methane. And then a very small fraction of that organic matter that was produced in the surface will become buried and sequestered on geological timescales. Okay, so both the water column and sediments are teeming with microbes that are using this organic matter as an electron donor. And they can couple it to different electron acceptors. And so this process of taking organic matter and using it as an electron donor, this is known as heterotrophy. So, they can use oxygen or sulfate or iron. And in doing so, they're able to, through breaking down these organic compounds, they're able to get energy for cellular function and carbon for building new biomass and new macromolecules. And the activity of these heterotrophic microbes is also really important for biogeochemical cycling. So they do this, they get energy and carbon, but in breaking down these compounds, they also release nutrients associated with the compound. So things like nitrogen, things like phosphorus, things like metals. And so these heterotrophic microbes in the ocean are really important for the recycling and the regeneration of nutrients. So before I start talking about microbial processes, I just want to talk a little bit about organic matter in the ocean. So organic matter is really this very complex soup, this mixture of thousands of individual compounds. In general, almost all of the organic compounds we find in the ocean come from biological photosynthesis. That could either be, from land, from the terrestrial biosphere, or from the marine biosphere. The primary source of organic matter to the ocean is typically, from phytoplankton, from the photosynthetic activity of organisms in the upper water column. But there's lots of terrestrial inputs. So, there's rivers, there's coastal erosions, there's lots of ways where organic matter from land can also enter these systems. And all of these compounds are continuously transformed, right, through both microbial processes, but through abiotic processes as well, like photodegradation. And so that really contributes to this complexity. So one of the things we've learned, over the last few decades of, lots and lots of studies of people looking at organic matter and thinking about its reactivity is that these compounds are not the same in terms of their bioavailability to microbes. And the bioavailability, you can think of how edible these compounds are, how readily organisms will take them up. So when we think about pools of organic matter, dissolved or particulate, or if we're thinking of sediments or the water column, generally, there's a pool of compounds that we consider to be labile that organisms will take up very quickly. So maybe on the order of days to weeks, or even faster sometimes if we're thinking about surface waters. And then most of the organic matter we think is recalcitrant. So this stuff is going to be more resistant to microbial degradation. And these compounds or these pools of organic matter can linger around in the environment for hundreds or even thousands of years. And so people have done work to try to understand sort of what controls the bioavailability of these different pools to microbes. And so there's generally, and again, this is sort of an oversimplification, but there's some general physiochemical properties that we think affects how easily microbes will take up a compound. So bioavailable compounds tend to sort of be smaller in size. They're very simple chemically, so they tend to be more aliphatic, and the C14 age is younger. And then on the other side of the spectrum, the more recalcitrant material tends to be, larger in size. It has a higher molecular weight. It tends to be more aromatic, and it's older in age. It's been hanging around the environment for a very long time. So, not all organic matter is the same. It's not all as equally bioavailable to microbes. And so one of the things that we wanted to investigate a few years ago is, okay, we know not all organic matter is the same, but what about the other side of this? What about the microbiology? Are all microbes the same when it comes to their capacity to break down organic matter? And if not, why? So we did these experiments where we took these different marine bacterial isolates. These were just very common isolates that were part of a culture collection of marine bacteria at MIT. And we could grow them up. They're very tractable. And we could incubate them with sediment, sterilized sediment. So we'd be killing off the natural community, but it would allow us to keep that natural mixture of organic matter. And then we would incubate them in the specialized bioreactor system that I'll talk about in a second. And it would allow us to evaluate what each species was eating and how much. And so we could try to understand sort of what the capacity of these two different marine bacteria, these two heterotrophs, is for degrading organic matter. So this is the system we incubated in. So Steve really downplays his contributions because when I joined the lab, Steve had already designed the system. And it sort of looked close to what it was. There was a lot of method development and tweaking that happened afterwards. But what the system allows us to do is this is a 3-liter culture vessel, and we can incubate any combination of microbes and organic carbon in there. And it's a flow-through system, so we can measure how much CO2 is being produced by the microbes. We can make these measurements every 15 seconds, 30 seconds, 60 seconds. And then we trap the CO2 in these molecular sieve traps. And then we purify them on a vacuum line, and we can send that purified CO2 for C13 and C14 analyses. And so this isotopic signature, these C13 and these C14 signatures of the CO2 that these microbes are breathing out, those are sort of like a chemical fingerprint, right? They can tell us what microbes might be consuming. And so if we, here we go. So this is based on the fact that these different carbon inputs that I just showed you, they're going to have different isotopic signatures, right? So these have different C13 signatures that can help us distinguish between different sources of organic matter in the sediment sample. And then if we bring in our C14 values, we can further resolve these sources, and we can figure out if microbes are eating compounds that are, recently photosynthesized, or something that's really old or anthropogenic, like if there's an oil seep or there's contaminants that have come into the system. Okay, so how does this help us understand microbes and what they're eating? This goes back to this very, well-known and old principle that some of you may already heard of, the you are what you eat, or in our case, you are what you breathe principle. So if you have a heterotrophic microbe here and you have a complex pool or a mixture of organic compounds, and our microbe here is going to degrade, organic matter from a specific pool or a specific type of substrate, then its biomass, so it's things like its lipids, its DNA, its protein, is going to show a similar isotopic signature as that substrate. But in our case, we know that CO2 respired from heterotrophs is also going to have a similar isotopic signature. So we can use the isotopic signature of CO2, so essentially what these microbes are breathing, as a natural tracer to figure out what they ate. So to sort of give you an idea, oh, sorry, keep forgetting I have to click twice. So that's to give you an idea of what this system does. And so with these experiments, as I mentioned, we had 2 different isolates. And I'm only going to show you a fraction of this data. This data has been published for a few years, so if you're interested, you can pull it up. But I'm only going to show you 2 of our plots. So we did this with 2 different types of sediments. This is a blue one and an orange one, so you can tell the difference. And they were both collected from a similar basin. They're both from Guaymas Basin, in case you're interested, that region. So our plots here, on the x-axis, we have time. So these incubations lasted between about 4 to 6 days. On the y-axis, we have our microbial respiration rate. And then on the secondary y-axis, we have cell counts. So every 12 hours or so, there's like a sampling port on the reactor. We can collect subsamples and we can do plate counts to track the growth of the organisms. And so what you see is that regardless of which sediment core we used and what type of sediment, this Pseudo-Alteromonas species had a higher respiration rate than the Vibrio. And interestingly, they both have a similar pattern where they sort of have this increase in CO2 and then it sort of tails off, sort of like a standard growth curve. But the Pseudo-Alteromonas has, a higher peak, so more CO2 respired, and the total amount of CO2 we collected compared to the Vibrio was about twice as much. And so what this tells us is that not all microbes are equal in terms of their capacity to degrade organic matter. And so the degradation or the cycling of organic matter is going to depend on the species that you have that are present and active in that environment. So one of the other things we did after we saw these sort of CO2 and isotopic patterns is to try to understand why. What is happening at a genomic level that can help us explain these patterns? And that's something that I'm very interested in, that we've tried to do in my lab, is to try to connect these biogeochemical observations with the actual mechanisms and with the actual genomic potential of the microbes that we work with. So with these 2 isolates, one of the really nice things is they have sequenced genomes, so we can go in and look at what genes and what enzymes they have. And so we went in and we looked for different extracellular enzymes or exoenzymes, which I'll talk about in a sec. But these are enzymes that are very important to accessing and breaking down organic matter. And what we see is that the Pseudo-Alteromonas species that, ate about twice as much organic matter based on our measurements, it has way more copy numbers of genes that encode for these peptidase enzymes that break down proteins. These are extracellular proteins that would be present in organic matter. And so this is a really nice sort of agreement between, our CO2 measurements, our isotopic values, and the genes that we suspect are important for accessing and breaking down organic matter. And it's really consistent with our broader understanding of how microbes break down and access organic matter. So this is really, a lot of this is based on the work of Carol Arnosti, who's really been a pioneer in measuring these enzymes in marine systems. But most organic matter in the ocean, like I told you, most of it is coming from phytoplankton. And most of that material is macromolecular. So what that means is that it's too big for cells to take up directly. So what some cells will do, what they've evolved to do, is they will make the enzymes to break down the organic matter. They will release this outside of their cells. And you can think of it like little scissors that chop up the material into smaller pieces. And then they can take up the smaller pieces. And so they're often referred to as primary degraders in the literature because they're the ones primarily breaking down the organic matter. And then we have this sort of secondary trophic level. So typically we have these two trophic levels when we think about organic matter degradation. There's these organisms that don't have exoenzymes that would rely on like metabolites, like sugars, amino acids, or maybe even some of the degradation products that the primary producers have released. And so these exoenzymes are really a key step in breaking down organic matter, and hence why when we do these experiments, when we have a species that has more gene copies for these exoenzymes, we actually are measuring a lot more organic matter being remineralized. So I'm going to tell you about one of the more recent studies we've done with the reactor. We demonstrated that these microbes have these differing capacities, but one of the other things I've always been interested in is thinking about how microbial interactions come into play when we think about organic matter turnover. So over the past few years, there's been a couple of studies that have begun to explore this in a really nice quantitative way. This is a study by Otto Cadero's group in 2018. And they did these co-culture incubations of this primary degrader that can break down chitin. So this is a complex polymer you can find in the ocean. And so they did these experiments where they took the Vibrio and they incubated it with the secondary degrader that they know doesn't have the enzymes. And what they found was that the secondary degrader actually ends up acting like a parasite, slowing down the overall degradation rate of the chitin. And then more recently, there was a paper in ISME using similar isolates from this culture collection, but using a microfluidics approach. And what they actually found was that the interactions between this primary degrader and the secondary degrader, it changed over time. So sometimes it was like symbiotic, sometimes it was parasitic. And so it was not the same depending on the stage of the growth. And so one of the things that we were interested in investigating was interactions, not between primary degraders and secondary degraders, but really interactions within the same trophic level. And specifically, we were interested in these primary degraders because those are the key organisms that have these enzymes that are going to break down the organic matter. So we did this, I went from a Mac to a PC. And it screwed up my cute little microbial cartoon. I paid an illustrator to make two bacteria as gladiators. So we called these experiments bacterial gladiators. We actually even had it in the title of the paper, but the editor asked us to take it out. They were like, it's not appropriate. So yeah, we have these two adorable little gladiators. And our goal was to incubate these guys. So, if you remember, these are the same isolates that we had incubated as monocultures. And so we already had this background data on what their respiration is like and what they're eating when they're by themselves. And we thought, okay, let's put them together in the bioreactor and see how they compete for different substrates and who's going to be the winner. One of the challenges in doing this work was that we couldn't figure out how to track who would win. because on plates they look identical. And so what we ended up having to do was to develop gene assays to be able to quantify each species. So we ended up developing these assays, and then once we did that, then it was very, very easy to do it. And this was work that was led by my PhD student, Sam McGill. And it's, again, I'm only going to show you like a slice of the data, but it's published now for almost 2 years in ISME. So if you're interested in it, then you can check it out. Okay, so we incubated our 2 isolates with these 3 different substrates. So we have glucosamine, which is a very simple sugar. This is usually what we grow them on when we want them to grow up really fast for incubations. Then on the opposite side, we have what I'm calling unimpacted marine sediment. And that's just like normal marine sediment from the bottom of the ocean. It's not like undergoing heating or doing anything weird. It's what like some scientists would just call normal, and Nagissa calls it boring sediment, just regular sediment. And so this would be mostly a mixture of complex substrates. And then we have the sediment that is has undergone hydrothermal heating. So it's been collected from an area where there's some low temperature heating. And what we know about low temperature heating and organic matter is that it can generate really labile organic compounds for microbes. So it can generate organic acids and other small substrates that are readily accessible for microbes. And so we were able to use these 3 different substrates to sort of see how the complexity of the substrates would affect the interactions between these 2 microbes. So again, these are our 2, the isolates. There's one, right? The Pseudoalteromonas has this advantage because it has more genome copies, but in theory, they should both be able to break down the organic matter. And again, we have all this background data. Okay, so who wins these gladiator competitions? It really depends on the substrate that we use. So when we incubated them with, oh, and I should mention again, sorry. So these are again data from the bioreactor. So we have microbial respiration rate on the y-axis, and on the x-axis we have time. And again, we have cell density on the secondary y-axis, and this time it's not plate counts, it's our gene assays. So when we incubate them with a simple substrate like this glucosamine, our Vibrio species actually outcompetes the Pseudoalteromonas, and it becomes really dominant throughout the incubation period. When they get incubated together in the presence of a more complex substrate that requires these exoenzymes, the Pseudoalteromonas appears to outgrow the Vibrio, but this is a very nuanced pattern. So for example, during the incubation with the hydrothermal sediment, the Pseudoalteromonas does much better, and there's sort of a big difference in cell density between the Vibrio and the Pseudoalteromonas. When we incubate them with the really complex stuff, so there's just not a lot of labile material around, these cell densities are a lot closer together. So the Pseudoalteromonas is still sort of dominating, but not as much as we see here. And so one of the things that's been hypothesized, not just in marine sediments, but thinking about interactions in the different environments, is that it's thought that when microbes are exposed to complex pools of substrate or complex organic matter, it can lead to the separation of degradative activities. So it can lead to these specialized niches, and so you can have the coexistence between species. So they're not directly competing with one another. And that's what we think is happening here, is we have more of a coexistence versus the sugar where, you know, one of them is just barely growing and the other one is doing really well. So the other thing we're really interested in is the actual turnover of organic matter and how that is shaped by the dynamics of these 2 species. So in these next few slides, I'm just going to focus on the sediments. I'm not going to focus on the glucosamine. And so as you remember, we have these background incubations. So, you know, we already know how much CO2 they respire out as monocultures. And what we really wanted to see was, is it going to be something like A plus B equals C? And the answer is it's not. It's actually more. When they're together, they seem to respire more, especially in the case of the hydrothermal sediment. Something really interesting happens there where they're eating or degrading a lot more organic matter than when they were grown as monocultures. And so we think this again supports this idea that when species are grown together with a complex pool of substrates, then you get this division of labor arising. And so they can differentiate into sort of these distinct niches, and you can get the simultaneous utilization of the organic matter pools, and then you get this overall increase in organic matter degradation. Okay, so the other thing we did during these incubations is we collected CO2 fractions of what these microbes were breathing out for isotopic analysis. And so here I am showing you the mass weighted average values for each incubation. So what we see is that when we incubate them together, the C14 values become more positive, right? So more C14 positive values are indicative of utilization of more recently photosynthesized carbon. And I didn't talk a lot about the sediments we use, but this is an environment where there's very productive overlying waters, and there's a lot of phytoplankton, and sort of it's just raining down phytoplankton biomass to the sea floor. And the C13 signatures, again, they become more positive when we incubate them as co-cultures compared to the monocultures. And so it's consistent with greater degradation of C13 enriched organic matter that you see coming in from phytoplankton. So that's actually all I'm going to say for the isotopes, but we have them over time, and we did these crazy mass balances over time to show how it changes. But it's in the paper, so if you're interested, please check it out. Okay, so to sort of summarize this data, what we see is that the interactions between microbes is influenced by the complexity of the carbon pool. And this is really interesting to think about, because in marine environments, the quality and the quantity of carbon actually changes. Like if you have a phytoplankton bloom, it can shift from labile to complex. And so this goes back to some of that earlier work I showed you with microfluidics that sort of shows how dynamic these interactions can be. But when we have labile substrate, it seems like there's more competition between the two species. They're competing for the exact same substrates. But when they're within sort of this complex pool where there's different pools of organic matter or different compounds, then you can get more cooperative interactions, and you can sort of get this simultaneous and sort of synergistic degradation of the carbon pool, because these different species can sort of attack and go after different types of organic matter. Okay, so now I'm going to switch gears a bit, and we're going to go back in time, 3 billion years, maybe even more. So I want to talk about some of these same processes, but in the context of ancient marine environments, and specifically thinking about how the capabilities of microbes to degrade organic matter, how that may have evolved over time. Okay, so I threw in this slide, so before we jump into the evolution of microbial heterotrophy, I threw in this slide about the evolution of Earth's surface environment, which is often thought about in the context of oxygen and our planet going from this anoxic world to this oxic world. I realized there might not be a lot of geology students in this department, so I figured I would just very quickly give you like 3 billion years and 30 seconds. So like, Greg, cover your ears, because I'm going to probably say things wrong or oversimplify them. But we think life emerged around like 3.7, 3.8 billion years ago. This is a time where the planet was completely dominated by anaerobes, right? We don't think there was any oxic organism really there. There's no oxic respiration. There's photosynthesis, but it's anoxygenic photosynthesis. Sometime around like 2.7, 2.8 billion years ago, depending on whose molecular clock or what evidence you believe, cyanobacteria appear. And then after a few 100 million years after they appear, we have what leads to this great oxidation event, which we believe cyanobacteria are responsible for. So we have this big increase in oxygen, maybe this super increase that some people have studied. And then we have sort of this higher, oxygen levels on the planet. They're not quite modern throughout the Proterozoic. Geologists actually call this the boring billion, which is really fun because eukaryotes evolved during this. And I'm like, it's a lot of fun microbial things evolving in life, but they say it's boring. And then, eventually you get to another increase in oxygen, and you get, plants and complex life. And then the planet starts looking to what we're used to. And there's some snowball Earth events in there too. So that's 3 billion years of history in one slide to give you some context. But some of the things that we still don't understand and what we were trying to think about when we were thinking about microbial heterotrophy is, this transition of going from this anoxic to this oxic world. People have studied cyanobacteria for a long time and thought about oxygenation in that context, but we were interested in thinking about the contribution of heterotrophic microbes. And one of the things we were thinking about is this question of when cyanobacteria evolved, why did they take so long? Why is there this lag between when they evolve and when oxygen accumulates? And what sort of supported this big transition or this jump over these few 100 million years? So one aspect that's been invoked in the literature is the availability of nutrients, right? So this is the equation for oxygenic photosynthesis. And we know that cyanobacteria require light, but they also require nutrients like iron, like phosphorus, and trace metals. And so in modern environments, we see this all the time when there's an input of nutrients. This is a satellite image of Lake Erie. When there's an input of nutrients, we have this bloom of cyanobacteria. And there's many, many papers. This is a recent one I pulled from 2022, where they tie the great oxidation event and that increase in oxygen and the increase of cyanobacteria, that's really what we're talking about, is increased O2 production, to the increased availability of phosphorus. And specifically, they suggest it's due to an increase in the recycling of phosphorus and the regeneration of phosphorus. And that would directly implicate heterotrophic microbes because they are the ones that are responsible for breaking down organic bound phosphorus or any organic bound nutrients and recycling and regenerating them both in the water column and the sediment. So going back to the marine carbon cycle, and thinking again about these exoenzymes in this context of sort of how nutrients and carbon flows through the water column and how it's buried. So, this phytoplankton biomass, most of it is going to contribute to what we call the particulate organic matter pool. But we know phytoplankton can also release organic compounds, typically large ones, what we would consider high molecular weight dissolved organic carbon. Our bacteria or our archaea in the water column, they can only access low molecular weight dissolved organic matter. That's what they can take up. So anything from these two pools that needs to sort of get accessed, or if these microbes want to access it, it has to either be done through photodegradation or through enzymatic hydrolysis. And so these exoenzymes, again, not only allow these microbes to really efficiently scavenge carbon and nutrients from the environment, but biogeochemically, they're really important because they're going to dictate how much of this biomass gets recycled and regenerated versus how much of it gets buried in sediments. So one of the things we've been trying to do is try to reconstruct the evolutionary history of these exoenzymes. This is very difficult because we don't have a time machine and there's no microbial fossils, right? We have very little data. So I'm showing you this figure of these different metabolisms. So the red are heterotrophic, the green are phototrophs. And, we have some idea of when these metabolisms appeared. I didn't even add fermentation on here because we have no constraint on that, but it's probably very early. But, we have the first cells appear, and the only information we have about when exoenzymes may have appeared is from a molecular clock paper that specifically dated genes related to chitin, right? So chitin is this very specific molecule associated with fungi and arthropods, and it suggests that bacteria acquired them around 670 million years ago. But between that and sort of, life evolving and the great oxidation event, we don't have a very good sense of how much exoenzymes were there, who had them, and how they could have influenced biogeochemical cycling. So one of the things we did is we first asked, are these exoenzymes ubiquitous across these metabolic groups? Because we know that the emergence and the ecological impact of these different metabolic groups would have varied over time. So what we did was we compiled publicly available genomes. This was led by my PhD student, Alia Sanger. We compiled both genomes from isolates and we compiled metagenomes. So there's tons and tons of data available for this online. And we searched for 23 known exoenzymes, and then we used these different bioinformatic tools to try to predict the localization of these enzymes, right? So it's not that these enzymes are really novel per se. Like, for example, a protease. Every cell has a protease. If the cell doesn't have a protease, it would die. It's the localization. It's the fact that these enzymes are directed to function outside of the cell. And so these tools will look for these signal peptide sequences and the general structure of the enzyme and give you a prediction of where they might be located. So we ended up with, well, we actually had 3,500 genomes. And then a reviewer said, that's not enough. So then we I know. So then we went back to, and we doubled it to 7,000 genomes, and our conclusions were exactly the same. But we took all of these genomes and we assigned them to different metabolisms based on marker genes. In the case of aerobic heterotrophy, we wanted this to be like a snapshot of sort of modern marine environments. And so we pulled a lot of data from the Terra Oceans database. And then for some of them, there's no marker genes. Like for fermentation, there's no marker genes, but there was a paper in 2023 that compiled a bunch of genomes, and so we could use it. And then we also added in phototrophs because, you know, I wanted to see if this mechanism was widespread among all microbes, and so we included them. Okay, so this is what we observe when we look for these exoenzymes across these thousands of genomes. So this is the percentage of individual genomes that contain exoenzymes. And again, we looked for like 23 of them, but we've collapsed them into carbohydrate acting and protein acting. And so what you can see is that they're not evenly distributed, right? So among, you know, aerobes, looking at aerobic heterotrophy, they have exoenzymes. That's not surprising. You know, people have measured this in modern environments. And among anaerobes, we see that fermenters have them, which again, people have studied fermenters and implicated fermenters in the degradation of organic matter in anoxic environments. But we also see them in ferric iron reduction, so in similatory iron reducers and also in nitrate reducers. And then very little, if not any, associated with sulfate reducers and methanogens. And we have some with phototrophs, not too much, but there are some there. And so when we saw this, and when I saw this, I was actually kind of relieved because some of these genomic tools, they will make predictions, and it's kind of like black magic. You don't actually know if they're making a correct prediction. But this was exciting because this matches what people have measured in the environment or in the lab. And that's always fantastic when we look at genomes, is if we can get things that sort of are consistent with actual observations. So this is a study I'm showing you from Jen Bowen's group. They did incubations with, these are sediments collected from Plum Island, which is like a long-term study site. And they added nitrate and they looked at organic matter degradation, and they found that nitrate actually stimulated organic matter degradation. And they also did FTIC-RMS analysis, and they suggest in their paper that it stimulates the degradation of more recalcitrant organic matter. And recalcitrant, we typically think of being more complex things that require exoenzymes to break down. This is another study. This is sort of thinking about it on the opposite end, right? We find nitrate reducers having these exoenzymes. We don't see them in sulfate reducers or methanogens. And this is a really beautiful study by Bo Barker's group. Oh, it's kind of weird here, but this is a really nice study where they did radio tracer experiments with Baltic Sea sediments. And they measured rates of organic matter degradation, and their conclusion was that the organic matter degradation rates don't vary in the sulfate or methane transition zones. And they suggest in their study, one of their conclusions is that these terminal steps are not exerting feedback on what they say is the upstream hydrolytic or fermentative processes. And so again, if these metabolisms or these microbes have exoenzymes, we should see really high rates of organic matter degradation or any rates of organic matter degradation, but we don't, because they likely are part of the secondary degraders that are relying on other organisms for substrates. So what accounts for the trends in our data? So we think this is related to bioenergetics and how cells invest energy and the energy that's available to them. So, you know, I showed you these different metabolisms. They are taking organic matter as an electron donor. They couple it to different electron acceptors. And this leads to differences in redox potential and the amount of free energy or the amount of ATP that's available to the cell. So this is like, you've probably seen some version of this. This is essentially the redox tower. So what we see is that in metabolisms that are using more energetically favorable electron acceptors like O2, like nitrate, they tend to have exoenzymes. And then ones that are sort of at the bottom of the tower, like sulfate reducers, methanogens, we see almost no exoenzymes with them. An exception to this is fermentation. Fermentation is different because it's not even doing anaerobic respiration. They generate energy through substrate level phosphorylation. And so it's very different. But one of the things we know about fermenters over the last sort of decade or so of research into them is that they actually, even though they're yielding less free energy of ATP through substrate level phosphorylation, they have really low catabolic power requirements. They don't have to maintain all of these, complex proteins for the anaerobic respiration. So they sort of have like a lower overhead of cellular energy, and they're actually more efficient at generating ATP. And so we think it's that they have this efficiency and this lower overhead that allows them to allocate a larger portion of their sort of energy budget towards producing exoenzymes. And this, I want to just say, is consistent with a lot of the work and a lot of the ideas that have been put forth by people that have been studying exoenzymes in soils. Like Steve Allison, this is a paper that he was a part of, and he wrote a fantastic book chapter on this called The Economics of Exoenzymes, where he goes into this in detail. But what he suggests and what we know is true is that secreting and producing these enzymes is costly, right? It requires carbon, it requires nitrogen, and it's costly because once you secrete the enzyme into the environment, it could float away. Maybe it could cleave something and you get a substrate back. Maybe a cheater could come eat it. So you're not necessarily guaranteed sort of a return on your investment. And so the fact that we see it in metabolisms that tend to be more sort of energetically rich because they're using more favorable electron acceptors sort of suggests that only organisms that already have energy will invest it into producing and secreting these exoenzymes. So my grad student said it's sort of like a rich get richer scenario. So if you already have enough energy, then you'll allocate some towards these enzymes and engage in this behavior where you may or may not get anything back out of it. Okay, so one of the other things that we did, is, I showed you that we looked at this across these different metabolic groups, and we found that, these exoenzymes are not uniformly distributed. So we also used molecular clocks to try to date the origin of them. And we specifically focused on alkaline phosphatase. I'm going to go through this a little bit quickly because I think I'm running out of time here. Alkaline phosphatase is really important for breaking down organophosphorus compounds. This is a study of alkaline phosphatase in the Terra Oceans database. So you can see some are located inside the cell, some are located outside the cell, some are extracellular, and they're encoded by 3 well-known genes. And so the other reason we looked at alkaline phosphatase is we know that when we think about early Earth, phosphorus has considered to be a very important limiting nutrient for early cyanobacteria. So we know that this would be a very important enzyme for regenerating nutrients that primary producers need. Okay, so in order to do this, I am not a molecular clock person. We collaborated with Rika Anderson and her postdoc Joe Bowden, and they had actually just published a molecular clock paper where they were trying to date the timing of different enzymes related to phosphorus cycling. They hadn't done alkaline phosphatase, so it was just a very happy accident. So they had created this tree of life. This is what they call their molecular clock. And we used this molecular clock to specifically look for the sequences for Pho A, Pho X, and Pho D. And then we did all sorts of other assumptions to assume which clades had extracellular ones and analyze them. And then we focused on 3 clades that we were sure had extracellular alkaline phosphatases, and then ran 3 different clock models. And this is what we got. So what I realized with these molecular clocks is not only are the error bars huge, but the different clocks can give you very different values, okay? So this might be like my first and last time doing molecular clock work. But what we see is that alkaline phosphatases, depending on the model and which model is better, and it seems to be like no consensus, is the extracellular alkaline phosphatases appeared sometime in the late Neoarchean or early Paleoproterozoic. And this window aligns sort of with the geochemical evidence we have with rising oxygen on Earth. Except we can't be sure if they emerged when there was these what we call whiffs of oxygen, or whether they evolved and emerged sort of when things had really kicked off and there was a lot more oxygen in the atmosphere. But what it does support is that after the Great Oxidation Event, or during the Great Oxidation Event, in the early Proterozoic oceans, there would have been this expansion of exoenzymes, and you would have had this boost in microbial phosphorus cycling. Okay, so I'm going to go through this extra quickly because I think I'm looking at the clock. Okay, so one of the other things we considered besides like just dating them and thinking about groups is what are the ecological conditions that would favor the selection and the evolution of these exoenzymes. So exoenzymes are very favorable under certain conditions, and they're not favorable under other conditions. So they're very favorable under diffusion limited settings. What that means is things like particles. And that's because if you're just a cell floating in the ocean and you release an exoenzyme, again, think about it, that enzyme is costly. If you just release it into the water, the chances of it actually interacting with a substrate or you getting that substrate back are pretty low. What we know happens both from modeling and experimental work in the lab is that if there's, say, a particle, microbes will sort of swim onto it, attach, and when there's a sufficient cell density and there's this diffusion limited environment where they can secrete the enzymes and there's a less likelihood that they will lose the enzyme or the substrate, then the community will sort of turn on their exoenzymes and everyone will produce it at once. So the availability of marine particles and the availability of, this diffusion limited setting would have been an important factor that would have favored exoenzymes. So to try to understand that, we did some modeling work. So we collaborated with someone at the University of Connecticut who had done a model looking at DOC over Earth history. And we sort of reconfigured the model to look at POC or POM as I'm showing you here. So what you see is that the amount of particulate organic matter or the amount of marine particles in the ocean has increased over time, right, relative to the modern. That makes sense because the amount of particulate organic matter is controlled by lots of factors, but a primary one is the amount of primary production you have, right? So if you have increased cyanobacterial growth, increased O2, you're going to have more particles in the ocean. And when we think about the amount of particulate organic matter to dissolved organic matter, what it tells us is that in the early Archean oceans, it was a very dissolved organic matter rich or DOM rich environment with not very many particles because we think primary production was very low in the Archean. And then after the Great Oxidation Event, when cyanobacteria have sort of taken over, there's a lot more oxygen around, you would have had a lot more particles, and you would have had a lot more ecological niches where the production of exoenzymes now becomes advantageous. So there's really this selective pressure in the environment to sort of access and break down these particles. Okay, so one of the other things we did is zoom out and not just consider the environment and these metabolic groups, but also the availability of the electron acceptors for these metabolic groups. So this is a diagram showing you these metabolisms we looked at. The purple ones had a lot of exoenzymes. These had almost none. And the widthness of that line tells you how ecologically important they were, right? So aerobic heterotrophs, they may have been around, but obviously they became more important after the Great Oxidation Event when there was oxygen around for them to use. Likewise, same with nitrate reducers. There was obviously more nitrate around after the Great Oxidation Event. And so what this tells us is before there was oxygen in the atmosphere, oxygen in the environment, most breakdown of organic matter would have relied solely on fermenters, right? Which are a fairly slow growing sort of sluggish metabolism. Yes, they're energy efficient, but they're not going to grow as fast as say like a nitrate reducer or an aerobic heterotroph. And so what we think this sort of led to was this sort of indirect positive feedback between primary producers and heterotrophs. So when you have sort of this pre-GOE ocean, you have very little particles. The only things that we know are likely around that have exoenzyme capability are iron reducers and fermenters. And so there's going to be very low recycling and limited release of these nutrients. You have really high carbon burial. We also compared this with the shale record, and we have that in the paper as well. And then after the great oxidation event where you have cyanobacteria releasing oxygen, that increases not only the availability of niches that would favor exoenzymes, but it starts putting out electron acceptors that are associated with these exoenzyme producing groups. So O2, nitrate, and so you get a lot more exoenzyme producing heterotrophs, which then break down more of this organic matter releasing phosphorus, nitrogen, trace metals. And so the more O2 they release, the happier the heterotrophs that produce exoenzymes are. So you get this enhanced nutrient cycling, which then gives you higher primary productivity and just furthers oxygen release. Okay, so these are my conclusions. There is a lot more work associated with this, but I put up a QR code of our preprint. So if you're interested in it, please check it out. But our sort of broad overall conclusions is that the redox state would have been a really important control, not only on microbial metabolisms, but on the efficiency of organic matter recycling. And so how much carbon gets buried, how much of those nutrients are regenerated, and that the rise of exoenzymes would have coincided with the rise of oxygen and really transformed organic matter turnover and nutrient cycling in the oceans. Okay, so with that, I just want to thank all of my collaborators. This is probably the most diverse group of collaborators I work with. There's like an astronomer, biochemical modelers, molecular clock people. So it's been a really fun project. And also, if you're interested in positions at McGill or in Canada, please reach out to me. So our graduate fellowship, like the equivalent of our NSF postdoctoral fellowship and our NSF graduate fellowships, they're now open to international students as of a year and a half ago, wink, wink. So if you are interested in coming to Canada or you're curious about it, please send me an email or reach out to me and I can send you that information. So thank you for listening and I'd be happy to answer any questions.
I think our first two questions are going to be in Jennifer. Either.
Okay. Thank you for your talk. It was pretty cool. I thought the slide on the mechanism behind, the microbes go around with particulate matter.
Yeah.
And you mentioned that there needs to be enough of them for them to turn on.
Yes.
The exoenzyme production.
Yeah.
I was wondering what the mechanism behind that was. How do they know?
How do they know that it's cell density controlled?
Yeah.
So that's a really good question. So I gave you this a lot of information into this one slide. We actually think it's controlled by quorum sensing. There's papers on that too. Ben Mouie's done these really nice studies where he's quantified quorum sensing molecules on marine particles, and there's really high concentrations of them. So we think Otto refers to it in his studies as this public goods behavior or this cooperative behavior where if you're just one, pretend you're just this one guy who swims over, you're not going to be able to eat this whole thing. You're probably not going to have a good chance at it. But when all your friends are around it and you guys all release your exoenzymes, you have a better chance of breaking it down. But also, if this enzyme that this organism releases, the substrate floats away, there's a really good chance somebody else might be able to get it. So it benefits the whole community. So if you pull up this paper by Ibrahimi 2018, it's in PNAS. They did these experiments in this modeling where they grew cells with chitin beads at different concentrations and found that there's actually a threshold. And once you reach that threshold, the particles get degraded. And if you don't reach that threshold, they will not degrade the particles. Yeah.
Is this cooperation interspecies or do they only want to cooperate with their own?
We don't know. That's a really good question. That's a fantastic question. We have no idea. In the natural environment, I assume that this is colonized by some diverse group of organisms. And like I mentioned, the Ben Mouie group, they extracted and analyzed the quorum sensing molecules, but we can't tell if those molecules are for a Vibrio to talk to a Vibrio or a Vibrio to talk to something else. We don't know. I assume it has to be across species because what are the chances of a single species colonizing it? But I don't know. Otto's group uses pure cultures, so they wouldn't have mixed, things. But that's a good question, and I just don't know a good answer to it.
Thank you.
Yeah.
Okay.
Go for it, Jen, sorry.
Hi, great talk. Kind of along the same lines, you mentioned that Pseudomonas had a higher respiration rate than
Yeah.
Vibrio. And this kind of aligns with Pseudomonas having substantially more number of genes encoding for exoenzymes.
Yeah.
I was just wondering if you've gone far along to do like some kind of transcriptomic or proteomic analysis, and what are the complexities of doing proteomics associated with exoenzymes given that they're being released?
This is a really good question, and I would say a very sensitive one for me, because when I did those incubations, I collected archives to do transcriptomics and proteomics, and I was like, okay, we'll do them. And I kept them from 2020 until recently in my freezer. So I moved them from Harvard and I brought them to my -80. And then I talked to a lot of people, and we could essentially do transcriptomics and figure out potentially if the exoenzyme is turned on. But in terms of actually like measuring a rate with proteomics, when I talked to people, they said it's really, really hard. And because our background is sediment, they were like, this is not like, you might get very mixed results with this. So I say it's very sensitive because in the summer we did a -80 now and we threw it all in the trash. I had to let it go. It's taking up a whole, like shelf on the -80. So the answer is we haven't done it. We want to do it, but in our case, I've just learned that when your background is sediment, you're very limited to what sort of analyses you can do. We could probably do transcriptomics, but the proteomics I don't think would work well. But it's definitely something we've thought about doing in the future. We have done not proteomics, but we have done enzyme measurements. This was actually a paper we did with Steve in 2017. So we did enzyme assays and we got activity rates, but we haven't done any metaproteomics. Yeah. Yeah.
Nice talking to you, son. So you early on you equate enzyme activity with gene counts.
Yeah.
Do you know that they actually reflect one another?
Like you're saying, are they all being expressed?
Right.
I have no idea. That is a really good question. I don't know. I guess the way we looked at it was like more of like a capacity. Do you know what I mean? So we're like,
Potential.
yeah, a potential. That's actually the term we use. Maybe you are a reviewer on this paper.
I did papers on hydrolytes.
Yes. So we use the word potential and we think of it as like the available toolkit. And we're like, yes, this is the potential, this is the toolkit, but as to whether everything's being turned on at once, I have no idea. Whether they're all being expressed, I don't know. That's a really good question, but yes, we think you're right. It's a potential, not like what they're exactly turning on at once.
But in some other experiments, it chlorogenic.
But that was on a mixed community.
Ah.
When Steve and I did that, we went to a salt pond, like outside of Woods Hole, and we took natural sediment. So we started off the reactor with looking at natural sediment before we did mixed individual isolates. And we put sediment in there and we just sort of watched what the microbes would do. And we did 16S sequencing, we did enzyme assays, we did cell counts. So we have enzyme assays, but then we don't have metagenomics to map it back onto. So yeah, that's something interesting we could definitely do and follow up on. But yeah, you're right. It's definitely potential, not like I don't know if they're turning them all on. Yeah. Yeah.
So the sentence here, production is favored by diffusion limited settings, is an intriguing thing to me. I work predominantly in sediment. So one of the things that characterizes the sediment pore space is that it's filled with mucus secretions, and that the diffusion through mucus is very different than through free solution because it acts as a polyelectrolyte and acts as a mesh work. And exoenzymes, the molecular weight of exoenzymes is in a region relative to mucus secretions where they should be inhibited, their diffusion should be inhibited through mucus. So at least most mucus.
Oh, interesting.
And so I'm wondering when the secretion of mucus evolved. in microorganisms.
I don't know, but can we collaborate and write a paper on this? I don't know. So when you say mucus, you mean EPS? Like, oh, okay, sorry. I thought you meant like animal mucus. I was like, I don't know, but we can find a molecular clock person to be able to help us. You know what? I don't know. I have no idea. Like, I assume that extracellular polysaccharides, it's like one of those very ancient things. You know, we say ancient. I don't know how you would time and date it. But so you're saying that when there's extracellular polysaccharides, it produces the diffusion of the enzymes?
Yeah.
Interesting.
Yeah, they don't move like that, moves through water, or a solvent.
But what about studies? So when I started digging into this, there's been like a lot of work on exoenzymes and like biofilms, right? People have done them on biofilms. And so in biofilms, they're really like a hot spot. So for me, actually, when I did this work, this was like the conundrum that would keep me up at night, is I'm like, is everything a mat on like, you know, a coastline, or were the oceans seeded with life? And the answer is like, nobody knows. So one of my co-authors was like, just write whatever you want in the paper, because we have no evidence. But I assume that in these sort of mat systems, which are enriched in EPS, they would be releasing those enzymes in there. And I feel like I read studies, but maybe interesting. I need to think about that some more.
Yeah. We'll be talking.
Yes, okay, yeah.
Talk more about
Yeah.
I mean, I was thinking of biofilms, because any kind of substrate you have.
Yeah.
And it could be as small as a particle. As soon as, you know, you get any bacteria,
Yeah.
they're secreting these exopolymers.
Yeah.
That then create an environment and well, very left, but typically the biofilms are a mixed culture. You don't just have one
Yeah.
type of bacteria.
Yeah,
They
yeah, I guess.
among each other.
Yeah, no, totally. So I assume that this is like, you get some sort of slime forming over this. The question is, how long do these guys interact for, right? Like, I've looked at sediment trap data, and some of these seem to get like degraded very, very quickly. Some are sinking. And again, it's one of those things where I was like, can I just find a number? And there's like so many numbers across so many different regions. I don't know how stable these biofilms are, if they're very long, a particle lasts for say hours to days or maybe longer. One other completely different side thing that we've gotten interested in with particles and thinking of these interactions is we're also thinking about horizontal gene transfer. I mentioned this to Gordon, is how these particles facilitate horizontal gene transfer, because they're considered hotspots for it. And so we are trying to spin up a project right now where we think about the abundance of marine particles in the ocean as being sort of a mediator of microbial evolution by facilitating horizontal gene transfer. Because these particles also allow organisms to do transformation, there's conjugations, there's all sorts of fascinating things that happen on there. So yeah, so maybe we can chat afterwards about this too.
I'll give you an NSF proposal that was beyond the frontiers, and you can resubmit it. That's what the panel decided, that this idea was beyond the frontiers.
I was gonna go to private foundation. So I don't know if you saw, but I'm in Canada, so they will never, NSERC will never fund work like this. So I went to the Heising Science Foundation for the exoenzyme stuff, and I'm hoping they will fund the horizontal gene transfer on particles too. Because yeah, in Canada, our funding agencies are even a little bit more conservative than I would say NSF in terms of basic versus applied. Yeah, yeah. Yeah.
So a facultative anaerobe would fit in more than one of your categories of metabolism. How did you handle that?
Oh, that's such a good question. We analyzed the markers across, I don't have the figure, but we analyzed the markers across the different metabolisms. And we have this figure in our supplemental information where something is potentially has a gene for nitrate and a gene for iron. And if I remember correctly, I think we put it in both just to be more on the conservative side. It wasn't as abundant as we actually thought it would be, but it definitely does exist where like something has a cytochrome to do nitrate reduction, but it can also maybe switch to sulfur. And that's one of the challenges with, I think, metagenomics is that it's great because you're like, oh, wow, we have all these genes. And then it raises more questions than answers because why do they have so many genes? It's not like they're running all these pathways. But yeah, we have a figure of that. And I believe we like counted them twice just to be on the safe side. Yeah.
All right, thank you so much for that.
Thank you.